Today we're going to be looking at organization of information on chromosomes. What have you learned in biology and about that?
DNA wraps around, what was it, histine?
Histones.
Histones to make up shapes. Let's see.
But that's more like the structure of the chromosome.
Oh, the organization?
Of information is going to be,
Oh.
you know, you get that 3-letter code.
Oh.
You've learned that. Which is basically the central dogma of how information flows, right? This, you've seen this, right? You can tell me if you haven't.
I didn't realize I'm on camera. Yeah.
You're not on camera, you can't see you,
Good thing.
hopefully. So, yeah, DNA serves as a template to replicate itself. It also serves as a template to make RNA copies, which carries the information to be translated by the ribosomes to produce proteins. So also, there's more than just, you know, proteins that it codes for. It codes for structural RNAs, tRNAs, rRNAs, small nuclear RNAs, and so forth. So, which is resulted, because of the RNAs, the coding for RNAs has resulted into our understanding of genes evolving, basically. Historically, a gene was something responsible for the characteristic or phenotype years ago, right? You ever see, like, you know, what was that guy's name? Not Mandel.
No.
Like with the flowers or the peas and the different colors. Yeah. But that was it resulted in the phenotype. Genes, you know, there's a gene for the colored pea or the white pea or so forth, and that was, you know, the genes. Then Beadle and Tatum, through the mutation studies, defined genes as something that could be coded for an enzyme, like one gene results in one enzyme. But now we understand that genes are defined as segments of DNA that code for peptides and RNA. Because, I mean, RNA is an important gene also. There can be different regulatory sequences that regulate the expression of these genes, and DNA can be expressed differently to yield different products. So one gene can yield several different products due to post-translational or processing of the message. So alternative splicing. Do you know about introns and exons?
So that's when the introns are arranged in different orders, or pieces not big at all?
Yeah, well, so you make the message, and the pre-message is going to contain all the introns and exons, but depending on, let's say, what cell it's in, there can be alternative splicing, where, like, you get, like, rather than, you might cut out part of an exon, or an exon, like you can have, let me get 2 colors. I bet you these don't work though. So you have, let's say the greens are the exons and the oranges are introns. So we said the greens are the exons, so the oranges are exons. That'll be another exon. Wait, greens are exons, oranges are introns. So we're going to cut out the oranges. You could have, I mean, this, let me find a different color now. You could have a scenario where this, oh no, gets spliced to this, and at the same time, in a different cell, it could be spliced directly to this. So, you know, we just cut out this one, or we can cut out this whole segment. And then based on that alternative splicing of those introns will result in 2 different forms of a protein, right? Because we're going to be lacking the amino acids coded for by that sequence there. Does that make sense?
Yeah, is this random or is this directed by something?
It's not random per se, so it's going to be directed most likely by microRNA sequences or small nuclear RNA sequences that'll be binding to that. If you could have a high concentration of a certain sequence of RNA in one cell versus another cell type, which could, you know, hide these splice sites perhaps. or do you understand?
Okay.
So, morning, how's it going? So we skipped to chapter 24.
Okay.
So the last exam will have the chapters like it says on the syllabus.
Okay.
Sound good?
Sounds good.
Okay. So we all know DNA is very large macromolecule. Linear dimension of DNA is much bigger than the virions or cells that contain in them, right? This is a picture of a virus, a phage, with all its DNA spilled out of its protein. Pretty cool shot, huh? So in this example, bacteriophage, the DNA itself, the bacteriophage itself is about 0.2 microns long and about 0.1 microns wide, whereas the DNA is about 60 micrometers in length. So all of this DNA has to fit into that little tiny protein package. So DNA in a virion or cell is organized into compact forms, typically via coiling and associated with proteins. And you were already discussing some of those proteins this morning, the histone proteins. And then it also, DNA will coil and supercoil. And it's amazing, we got the phone here. Yeah. Do you guys ever use phones like this?
My old job, baby.
You ever notice how these get twisted and you can actually, I don't know if you can, they'll start to form those supercoils basically. You see they start forming coils upon the coils, and you can really start to make them compact, which really probably isn't good for the phone. But it's one way that we get the coils packed it up. Molecular coding of protein sequence information and transcription. One-stranded, double-stranded DNA acts as a molecular template for the RNA synthesis. So we have the DNA acts as a template for the messenger RNA, and then there's that 3-nucleotide codon which provides information to translate the code of the RNA into what amino acid is going to be added on that particular protein, right? In translation, triplets of nucleotides in the mRNA bind to the complementary triplets in our tRNAs, or the transfer RNAs. And then the tRNA molecules contain amino acids associated with that particular triplet. Amino acids are thus assembled into the peptides, and the protein sequence determines its biological function and how it's going to fold and interact with other things. So this should all be stuff you guys have seen before, I'm sure. So you have DNA acts as a template for the RNA, and the 3 code sequence codes for individual amino acids. And it's always listed 5' to 3', and amino-terminus to carboxy-terminus. So how many genes per organism do we have? So E. coli has one singular circular chromosome. That's about 4.6 million base pairs, containing about 4,300 genes for proteins and 157 genes for catalytic or structural RNAs. Humans will have 24 discrete chromosomes, not circular. They're chromosomes. 3.1 billion base pairs and about 25,000 genes. So we got some more genes, but in a massively more amount of DNA though, right? 3.1 billion base pairs. Viruses, a lot of viruses have, only RNA or DNA for their genome, surrounded by a protein coat, and they'll use the host genes when they infect the plant, animal, or bacteria. We know that to be able to replicate themselves. RNA genomes may be as small as, small and single-stranded, HIV is single-stranded RNA, that's about 9,000 base pairs. Phage, about 4,220 base pairs. There's other, I think, hepatitis D virus I think has the, if I remember correct, the smallest genome for a virus. Of course, it can't replicate if it's the only virus to infect. It's one of the viruses that needs a helper virus to replicate.
I think that would make it less prevalent.
It is a lot less prevalent. One of the helper viruses, I think, is the hepatitis B virus. So when you find hepatitis D, most of the people who have hepatitis D have hepatitis B, I believe. So, because that hepatitis D virus relies on the hepatitis B virus for its own replication. And it increases the chances of, I think, liver cancer if you have both infections. Genomes may change form from circular to linear, et cetera, during the life cycle of different viruses. Bacterial genomes are double-stranded, circular DNA. And in E. coli, it's about 4.7 million base pairs, 850 times longer than the cell itself. They also contain extrachromosomal DNA. And it tells you what they are. I was going to ask you. But you've heard of plasmid DNA before. One of the big tools in molecular biology and biotechnology, plasmid DNA. Extrachromosomal, so it's not part of the main chromosome. It's smaller, about 2,000 to 10,000 base pairs. Swapped easily between bacteria, so that's how, if you have a chromosome that imparts one bacteria with antibiotic resistance, it can share it with other bacteria. And you can have a whole colony of bacteria that has antibiotic resistance after that.
Coli?
Huh?
Coli?
I forget the structural. Is it the pili that they connect together and it goes through? I haven't had that micro class since I was an undergrad, I guess. So plasmids, no essential genes, but often encode genes that degrade antibiotics, like I just said. And they exchange is one way bacteria acquire antibiotic resistance. And then the sizes of DNA and viral particles for some bacteria viruses or bacteriophage. So these are all viruses. And you go from 5,386 to T4 phage is 168,000 base pairs. So pretty large. And this is a picture showing you basically E. coli with its genome spilled out of its guts. So you can see, and then this is a representation of the E. coli cell and the E. coli DNA, how huge it is compared to the cell. So you got to fit all of this inside this little tiny green dot up there, if you can see. Eukaryotes like us, our DNA is in multiple discrete chromosomes, right? Number varies between species. Our cells, somatic cells, have 46 chromosomes. So we have 22 pairs of each chromosome, plus we have our sex chromosomes, the X and the Ys. We'll have a pair of those, either XX or XY, right? And the amount amounts to about 2 meters of DNA that we have in each of our cells. The length varies between chromosomes. This shows you chromatin, compacted chromatin, what it looks like. Typically when cells are about to divide. Mitochondria and chloroplasts also have DNA, their own separate genomes, and they're double-stranded circular DNA, similar to that of bacteria. Human mitochondrial DNA is about 16,000 base pairs, 2 to 10 mitochondrial DNAs per mitochondria. Plant mitochondrial DNA is about 200 to 2.5 million base pairs, 200,000 to 2.5 million. Chloroplasts about 120,000 to 160,000. So you get the mitochondrial proteins are produced and chloroplasts will produce their own proteins. But then there's also proteins produced in the nucleus of these cells that are incorporated in the mitochondria or chloroplast also. So DNA, chromosomes, genes, and complexity. Neither the total length of DNA nor the number of chromosomes correlate strongly with the complexity of an organism. I think there's some plants out there that have the most DNA per cell. Did they talk about? They have more genes than humans. Plants have way more genes than humans. Dogs and coyotes have 78 chromosomes. And they have a really strong ability to change their, basically mutate and change their structure and function, right? With all the dog breeding, they're real akin to that. Amphibians also have much more DNA to humans, and then the correlation between genome size and complexity is really poor because the vast majority of eukaryotic DNA is non-coding. What is it there for? We're not sure. Recent experimental work by Craig Ventnor suggested that living organisms could get by with less than 400 genes. But we got this vast amount of non-coding DNA. Some of it is regulatory, so to regulate, what genes are being on and off in different cells, and then others of it is just random, and they think it may result from historical viral infections, viral DNA that is incorporated into our genome. Viral genomic DNA may be associated with capsid proteins. Right, so DNA is packaged within proteins. Prokaryotic DNA is associated with proteins in the nucleoid. And then our DNA is organized with proteins in complexes called chromatin. So if you remember, we just said a small fraction of our total genome, human genome encodes for proteins, only about 1.5%. Like I was just talking about, we're not entirely clear about what the biological significance of the non-coding sequences. Some DNA regions directly participate in the regulation of gene expression. So we got promoter regions, which are regions where the RNA polymerase will start coding transcripts. And then there's regions also that can be way upstream or downstream of promoters that act as activators or inhibitors that regulate which promoters are going to be active. Termination signals, etc. Some DNA encodes for small regulatory RNA with poorly understood functions. And this, before I started this job, this was a real new hot area, looking at these small regulatory RNAs that regulates basically gene expression and which proteins are going to be produced and how much, or which variants of proteins are produced by altering the, what's it called? I said it earlier. My words have lost me. Alternative, transcript processing, RNA processing. Some DNA may just be junk and pieces of unwanted genes or remnants of viral infections from years ago. So, but eukaryotic genes, we know we have exons, and in this case I had the green as exons, and introns are the intervening sequences. It's one way, if you have trouble remembering which ones get cleaved out or are non-coding, or the introns. And both exons and introns get transcribed in the RNA, and you have this pre-mRNA that have to get the introns, cleaved out. And here's a couple examples of some introns and exons in, for example, ovalbumin. We have A, B, C, D, E, F, and G. All of these are introns that will get cleaved off. The only coding sequences here are the L 1, 2 through 7. And so you can see the vast majority of the initial transcript that made gets cut out. Introns are removed after the transcription, and the exon mRNA sequences are spliced together, and it creates a mature message RNA, mature transcript. There's other processing too that we'll learn about in the later chapter. Before, we never really believed that bacteria had introns also, but it turns out they do have introns in their genome. So, but about 25% of the sequenced bacterial genomes show the presence of introns. And they do not interrupt protein coding sequences, they interrupt mainly the tRNA sequences. So you have multiple tRNAs on one transcript, and they'll be cleaved out or on that transcript. Introns and phage genomes within the bacteria interrupt protein coding sequences. And many bacterial introns encode catalytic RNA sequences. What does that mean to you, a catalytic RNA sequence?
Speed something up.
So it's coding for an RNA molecule that has catalytic activity. So it's a ribozyme of some sort. So it's going to have some sort of catalytic activity is the RNA that it's going to have. So the bacterial introns will have activity. They have the ability to insert and reverse transcribe themselves into the genome and make DNA. So it's a way of altering the genome also. Which comes to transposons, which we'll find in eukaryotic genomes. And they're areas of the genome that are mobile. They transpose from one place in a chromosome to another, maybe even another chromosome. And they're called the jumping genes. Have you heard of that before? I think, what was her name, McClintock? Remember, people used to make fun of her. She studied corn. People scoffed at this idea back in the 60s or whatever, that these genes could move around. And then she ended up winning the Nobel Prize years and years later because of it, I think. So the transposons, the ends, they'll have these terminal repeats. Similar to like a lot of viruses will have terminal repeats. And these repeats can hybridize with complementary regions on the target DNA during insertion. So you'll have terminal repeats on the piece that's going in, and it'll get inserted there. Or when it comes out, both the region that gets inserted and the region that comes out will have these terminal repeats, and it'll be able to hybridize with insertion regions in the chromosome. So it's like a place where those genes will go into and out of. And transposons will account for almost 50% of our whole genome. So it's not, the genome's not like a static thing, right? So it's constantly able to change, right? Eukaryotes also contain highly repetitive DNA or simple sequence repeats, SSR. And these are short sequences, about 10 base pairs or less, repeated millions of times, and also known as satellite DNA. I think if you take Brigida's class, she has you look at what satellite DNA you have through some, I think it's molecular diagnostics or molecular biology. I can't remember which one she does it in. Because when the fragments or centrifuges, they call it satellite, the DNA separates into these discrete satellite band, and it's just these short sequences that are repeated over and over and again. They're associated with the centromeres and telomeres. You guys know what the centromeres and telomeres are? Yes, good. See, this is like a nice review chapter, not right? Huh? So telomeres, because eukaryotic chromosomes are not circular like bacterial chromosomes, we have these ends that form these special loop structures to keep DNA ends from unraveling. They contain multiple repeats with this general sequence, TX times GX times, and that's repeated over and over and over again. Or AXCY, they base pair with each other, where N is about 1,500 in mammals, and it's added not by DNA polymerase, it's added by telomerase. So, and why would we need something other than DNA polymerase to add these telomeres to the end of our chromosomes? Huh?
Ask that question again.
Why would we need something other than DNA telomerase to add these repeated units over and over again? You got any guesses? You probably.
They fall off. We need to add more.
Why did the telomeres fall off, or how did they? Well, yeah, that's what well, yeah, we add an RNA primer, right, when we duplicate, because you need to have a primer. We have the DNA here. This is the end of a chromosome. You need a template, right? And then you're going to be adding things to this. If this is the 3 prime, this would be the 5 prime. So both of these, if this is the other side of this, where are we going to get this primer? You know what I'm saying? Like this, you'll be able to make it to the end, pretty much. But here, we're going to need something to add on to make from there, to copy this into the sequence. So every time the cell divides, this will get shorter because there's no template there. This is where the enzyme telomerase comes in. It carries its own internal RNA sequence that it copies, it uses as a template to add bases to the ends of these. And that's why it's repeated over and over and over again of the same sequences. It's because of the sequence of the RNA portion of the telomerase acts as a template. I think we cover that in a later chapter in more detail, but I think this is more detail than this chapter covers it. But we'll see it in more detail. And telomeres are associated with cellular aging because every time the cell divides, it's going to be shortened after each round of replication. Thus, the cellular age of DNA, the cellular DNA will age, it will get shorter and shorter. And eventually, as it gets shorter and shorter, you'll end up shortening the chromosomes into DNA or protein coding regions that are required for that cell to maintain life. And then they'll lose their ability to divide after that, and they call that a Hayflick limit. But telomerase, if you overexpress it, you can get past that, which is how, like cancer cells.
Immortal lines.
Yeah, have immortal type cell lines, and they'll just boost the length of those telomeres. Centromeres are sequences which the proteins attach during mitosis. We see that. It's where the 2 daughter chromosomes are held together during mitosis, and then after DNA replication, but before the cell division. And it's essential for equal distribution of the chromosome sets to the daughter cells. And they have AT-rich repeated sequences of about 130 base pairs. And this shows an image of basically what we see with the telomeres and centromeres in a yeast chromosome, with telomeres at the end, and the centromere somewhere in the middle where the chromatin are held together during division. So, we have our supercoiling. So DNA is supercoiled because it's that huge length we have to pack into such a tiny space. Packing a large amount of DNAs within the cells, and we have to have access to that DNA also. We have to be able to read the information in the DNA sequence. There's several levels of organization, of which one of is supercoiling of the double-stranded helix. And that's basically what a supercoil is. So this would be your coil, like your double-stranded DNA. When that gets coiled upon itself, it's known as supercoiling. Just like I showed you with the phone cord. So a lot of people these days never used one of these, the younger kids. But I remember in my house when we had one, the thing would always supercoil itself and get all tangled up. It was such a pain in the... Yeah. So, supercoiling is the coiling of a coil. Non-supercoiled DNA is called relaxed. And if you ever worked with plasmid DNA, when you purify it, there's typically the 3 forms. You have relaxed or open circular relaxed DNA, supercoiled DNA, which runs fairly fast, or this is your relaxed circular, or linear is when you have a complete break that linearizes that plasmid. So supercoiling is a great influence on transcription and replication of DNA, right? Because if it's supercoiled, you don't have as much access to be able to replicate it. And it can be highly regulated, the supercoiling. And this is what you would typically see if you did a plasmid prep. A lot of times you won't see this single-stranded circular, but you will see supercoiled, generally is the majority of their plasmid DNA, and it'll run faster than the linear. Linear runs like, it's linear, so it'll separate based on its size. Supercoiled is well compacted, so it'll run faster through the agarose. And then the open circular runs a little bit slower because it's impeded by having that circle there. So supercoiling is part of transcription and translation, because in order to get transcription or translation, transcription or replication, you got to separate those two strands, the double strands of DNA, and when you do that, it'll cause DNA to supercoil in front, and then you find underwound DNA behind it. So it'll cause it to wind as you're trying to pull this. This would be known as a transcription bubble, where you've melted the two and you're making a new RNA here. It kind of overwinds the DNA ahead of it and underwinds behind it, and that adds like a force impeding the movement of this RNA polymerase or DNA polymerase also. So it'll slow down the movement, but there's enzymes that can relieve that stress or unwind that DNA. So normal B form relaxed DNA is about 10.5 base pairs per turn. Closed circular DNA is rarely relaxed. Strain induces supercoiling, and the strain is due to fewer helical turns, underwinding, and then the underwinding makes later separation of the strands easier. Linear DNA is underwound with the help of proteins to prevent strands from rotating. And this next image shows you different levels of supercoiling. You have complete circular, unwound, relaxed DNA. You get some coils, get some more, and you can get highly supercoiled DNA like that. And this is the effects of DNA underwinding. Your relaxed DNA, you have about 8 turns. Strained is going to have 7, and then your supercoil. And then if you have strand separation, it causes that underwinding, overwinding to occur. You can think of DNA as going to have twists and writhes. Like a straight ribbon, like relaxed DNA, looks like this. If you have a large writhe, you have small changes in the twist. You don't have the twist so much, but you have these writhes that form those big like loops. Zero writhes with large change will result in large change in that twist, kind of. So underwinding also will facilitate additional DNA structural changes, help maintain cruciforms and palindromes. What's a palindrome? Do you guys know?
It's a pattern that the first half mirrors the second half.
Yeah, like race car, right? So, yeah, the same thing with palindromic sequences of DNA. The first half mirrors the second, which can result in these cruciform type DNAs, because not only will it base pair, these will base pair with itself to form like a linear, it'll base pair onto itself to form cruciform DNA. Cruciforms will rarely occur in relaxed DNA, but it facilitates the formation of stretches of left-handed Z form of DNA. If you guys remember way back from that other chapter, you can have the Z form, B form, or there's the A form of DNA. Most common is B, but Z form gives you a left-handed twist that you can find there, like that. Topoisomers are DNAs that differ with what's known as a linking number. The same number of base pairs, same sequence, but different degree of supercoiling. And the conversion between topoisomers requires a DNA strand break. So there's topoisomerase, are enzymes that will do this. And the negatively supercoiled DNA is more compact, travels faster in agarose gel electrophoresis experiments than relaxed or nicked DNA. We saw that from before. So the supercoiled travels faster than the relaxed or nicked DNA, and the linear also. And then these are different topoisomers, so they have the different degrees of supercoiling based on what you can say your linking number. You have a decreasing linking number causes increase in, how fast that piece of DNA can migrate in an agarose gel. So this is your highly supercoiled, relaxed DNA, and different degrees of linking. And they do this by incubating for different amounts of time with topoisomerase. Topoisomerases are enzymes that change this linking number. And there's two major types. So there's type 1, which makes a transient cut in one DNA strand, just makes one strand and it allows the DNA to unwind around that one strand. Or type 2, which makes a transient cut in both strands. So it cuts both strands and then actually forms a covalent intermediate with the enzyme, and the DNA will unwind, and then it changes the links in steps of 2, but then it reforms that phosphodiester backbone. Topoisomerases 1 through 4 of E. coli, Topo 1 and Topo 3 are both type 1s, and it removes negative supertoils to relax DNA using single-stranded breaks. Topo 2 is a DNA gyrase. So we'll see more detail on that when we talk about replication, transcription. It introduces supercoils, uses ATP, and causes a double-stranded break. And this is the type 1 type topoisomerases. And typically there's an active site tyrosine that's involved in forming that single-stranded break. So in an open conformation, where's step 1? There is no step 1 here. This is just half of the figure. So the tyrosine here will come in and attack the double-stranded DNA, make a single-stranded break, and then it'll allow the other strand the unbroken strand passes through the break of the first strand, so it'll be able to unwind that way. And then eventually the enzyme is in a closed conformation, liberated. This 3' that was cleaved before just attacks that tyrosine that's covalently bound with the rest of that strand and reforms that DNA linkage. Sorry, I'm missing the other half of the figure, I guess. And this actually, have you ever heard of those Topo TA kits from Invitrogen or Life Technologies? So they're super efficient plasmids for cloning plasmids that they sell. And what they do is they come with the topoisomerase type 1 bound to the plasmid in this form, with this intermediate form where the topoisomerase is attached via its tyrosine on the plasmid, on both ends of the plasmid. So the insertion site, you just have to have the insert, and the enzyme enzymatically cleaves it together, so you don't need DNA ligase to ligase your insert into that plasmid. You use the topoisomerase to do that, and it's way more efficient than using DNA ligase. It's just a lot more expensive to buy because you have to make the topoisomerase and then add it to the plasmid and purify that topoisomerase plasmid intermediate before it links it back together, right? And they use a topoisomerase from the vaccinovirus for that kit. So, eukaryotic topoisomerases include Topo 1, 2 alpha, 2 beta, and 4. Topo 1 and 3 are type 1, just like in E. coli. Topo 2 isomerase includes 2 subfamilies, 2A and 2B, and they can relax both positive and negative supercoils. And this is the mechanism. Looks like we got it all here. Mechanism of eukaryotic type 2A topoisomerase. So in the first step, the multi-subunit enzyme binds to the segment of light blue of DNA molecule. So this is the C gate. The next step, the second segment of DNA molecule bound at the N gate. And you can see you can get a conformational change where in step 3, what do they say? The second segment of DNA is trapped by that N gate, and then both 5' hydroxy strands are broken, and then this requires ATP. You get passage of the DNA strand that wasn't broken through the other strand. It's passed through the break, and then you get religation here. The broken DNA is religated, and the second segment is released through the C gate. You release the ADP and phosphate, and then that can repeat itself, and you can get that release of supercoiling over and over. But that's an ATP-dependent process. If you notice, topo type 1s don't require ATP at all. So topoisomerases are targets for antibiotics, coumarins, and so forth. Inhibit bacterial type 1 topoisomerase from binding ATP. And if you inhibit that type 1, you'll block, transcription and replication, because you need to be able to unwind those supercoils to form that transcription or replication bubble, and then you'll block transcription translation that way. Quinolones inhibit the last step, which is the resealing of the DNA strand break. So you end up breaking those DNAs, you can't reseal them. Again, that's a good way to kill off bacteria. They're wide spectrum and mostly selective for bacterial enzymes. Mostly selective, right? So if they're not 100% selective, you might see side effects where, that's why sometimes you feel sick when you eat antibiotics. Also probably because you are sick, the reason you're eating antibiotics, right? Or eating antibiotics. What kind of person am I? So topoisomerase inhibitors used as chemotherapy agents also, right? Because you have to be able to unwind supercoils to copy the DNA. So cancer cells are replicating the most, so if you block their topoisomerases, cancer cells won't be able to replicate as well. Neither will your gut cells and your hair cells that replicate also, which is another reason why chemotherapeutics cause you to be sick, right? So it targets cancer because most rapidly growing, express topoisomerases. Eukaryotic type 1 topoisomerase inhibitors include camptothecin, irinotecan, and topotecan. Trap the enzyme DNA complex in its cleaved site. And type 2 topoisomerases include doxorubicin and eptoposide. And things you don't want to have to take. So supercoiled DNA forms plectonomic or toroid solenoid type structures. And we see plectonomic seen is in plasmids, involves right-handed superhelix with terminal loops. You'll get these superhelices and you form these big terminal loops. And this is like your plectotomic loop. Soloids we see in chromatin. Did you see pictures like this where you have these circular structures that are wrapped around? Well, I'll show you in a minute. So this is plectotomic, and these are the solenoids. Used in chromatin, involves tight left-handed turns and can resemble a garden hose on a reel, and provides more compaction. We got pictures of the compaction here. So eukaryotic chromosomal structure changes over the course of the cell cycle, right? So when you're non-dividing, it's the GO state. It's going to be different from interphase, gap1, G1, synthesis, S, and gap2 or G2 phase. Chromatin's amorphous, randomly dispersed. When you're preparing for division, or prophase, the chromosomes will become condensed in pairs of sister chromatins to form. Right? So you're replicating the DNA at this point and packaging those chromosomes and pairing them up so they can split. And then you get, so here's where you get the condensation of the chromosomes during prophase and the prophase, metaphase, anaphase, and telophase. You have the different, packaged of chromosomes. So the chromatin will consist of fibers of protein and DNA and small amounts of RNA. The DNA associates tightly with proteins called histones, right? Small proteins with lots of basic residues. Why would you have lots of basic residues in DNA-associated proteins? Any ideas? DNA is what? What does that stand for?
Deoxyribonucleic acid.
So it's an acid. Carries a negative charge. Basic residues often are positively charged because they accept protons. So you have positively charged proteins are going to be attracted to the negative charge on DNA. So you get tight binding between the two. Histones are also modified by methylation, acetylation, ADP ribosylation, phosphorylation, glycosylation, simulation, and ubiquitination. So they're heavily modified, or they can be heavily modified, which is going to modify how those chromosomes pack also. So all of these modifications are done by enzymes that will regulate the chromosome packaging. DNA and protein are packed into discrete units called nucleosomes, right? Nucleosomes basically consist of DNA wrapped around histones. So DNA length of 10 to the 5 micrometer, what's that, 10 to the 10,000 or 100,000? 100,000 micrometers. Fits into the nucleus that's about 5 to 10 micrometers. Partial unfolding reveals beads on a string. Like you were saying earlier, you have these beads on a string, and the beads are all 146 base pairs of DNA wrapped around 8 histones. The core nucleosome basically forms left-handed solenoid. The string is a linker DNA of 54 base pairs bound to histone 1, H1. The amino-terminal tails of histones stick out, which form sites for valent modification. That's where you get your acetylation, or ADP ribosylation, all the methylation, all those other things, and form important contacts between the nucleosomes. So the way you modify those tails will modify the contacts between the nucleosomes. So here you have a histone core, and this is a micrograph of one, electron micrograph. The nucleosomal DNA is underwound. Wrapping DNA around the histone core requires removal of one helical turn. And the underwinding occurs without a strand break, so it's compensatory positive supercoil forms. That supercoil is relaxed by a topoisomerase, leaves the DNA with a net negative supercoil. And this shows you their general structure. You got your 8 histone cores, and the DNA gets wrapped around it like that. And this next picture shows you, crystal structure with the histone core. You can see the alpha helices of each histone there with the DNA, double-stranded DNA wrapped around it. I always like pictures like that. The next one shows you those N-terminal tails of the histone, and this is where you get interactions between the nucleosomes, and you get your modifications on your histone tails, which will alter how those histones interact with each other, altering packaging of those chromosomes. This one shows you, again, this is a cartoon, so it might be easier to see than the crystal structures. But you get the tails, amino-terminal tails interacting with each other, and a histone octamer there. So histone binding depends on the DNA sequence. It's not random, occurs more often in AT-rich regions, staggering AA, AT, or TT at 10 base pair intervals, phased with the pitch of a helix, narrows the minor groove, bends the DNA. So you're bending the DNA, it makes that narrow, the minor groove. Do you remember how there's a major groove and a minor groove on the DNA? And it bends the DNA, facilitating that binding around that histone core. I don't know if you can see it on these. It's showing you the 2 or more AT is where you get the contact with the histone core. 2 or more AT again, and so forth. Nucleosomes will then assemble into a higher order structure. So the nucleosome formation compacts the DNA 7-fold, but overall compaction's still greater than 10,000-fold. So this is just the first step, is forming the nucleosomes. The next level structure is known as the 30 nanometer fiber. It occurs in regions where the sequence-specific non-histone proteins bind. 2 models in these figures, the solenoid model and the zig-zap model. And this is the 2 models they show. The solenoid model and your zig-zap. You gotta love that name, zig-zap model. Basically 2. And it's called the 30 nanometer fiber because they're about 30 nanometers thick. And it's basically coil of your histone curves. And the zig-zap is 1-start or 2-start helix. The solenoid is a 1-start helix. Then that gets folded even more. Higher order structures are not well understood, but it may vary between chromosome regions and even moments in time. Involves a loop of DNA, maybe with related genes, associated with the scaffold proteins. Includes Topo2 and SMC, or structural maintenance of chromosomes proteins. And you can see the chromosome scaffold here with the DNA loops coming off of these 30 nanometer fibers. You can see how the DNA is quite densely packed against that scaffold. And this figure shows you the overall compaction of eukaryotic chromosome. Normal DNA helix packed into a nucleosome, which then forms a 30 nanometer fiber, and these 30 nanometer fibers can be an extended form of a chromosome, and then they can form these loops on a condensed section that you'll see with the scaffold protein. And here's a nice one of these again. This is on. How do I get over there?
One human cell contains about 2 meters of DNA packed into 46 chromosomes, which fits inside a nucleus only 0.006 millimeters in diameter. How is this possible? A single very long DNA molecule is packed into one eukaryotic chromosome by progressive layers of coiling. First, DNA wraps around histone octamers to form a chain of bead-like nucleosomes. In this form, the DNA strand is compacted about 7-fold in length. In chromosomes isolated by very gentle methods, nucleosome cores are found organized into a higher order structure stabilized by histone H1 binding. The higher order structure, called the 30 nanometer fiber, provides an approximately 100-fold compaction of the DNA. The higher levels of folding are not yet understood, but certain regions of DNA seem to associate with a nuclear scaffold. The intervening regions form loops. In addition to other proteins, the scaffold contains large amounts of histone H1, which is also located in the interior of the 30 nanometer fiber. In this model, the many rosettes are further twisted into coils. Each chromatid is made up of many such coils. The final densely compacted chromosome is found during the mitotic phase of the cell cycle. The compaction of DNA in a mitotic chromosome is greater than 10,000-fold.
So a condensed chromosome structure is maintained by the SMC, scaffold maintenance chromosome proteins. And they have 5 domains, and N and C domains forms ATP binding sites. There's 2 major types, cohesions and condensins. The cohesions will help link sister chromatins, and the condensins help chromosomes to condense by creating positive supercoils. And this shows you some of the structures of the SMC proteins, where you have these SMC cohesions and condensins. So cohesions and condensions. Condensions help them condense. Cohesions bring those 2 sister chromatins together. So bacterial DNA is organized into nucleoids, and they can occupy much of the cell volume. Remember, they don't have the nucleus, but they have a nucleoid. DNA is attached to the plasma membrane, and scaffold-like structure organizes the circular DNA into 500 looped domains. DNA binds to proteins transiently, and they look similar as the rosettes that we see when they're dividing, that we see, the same rosettes that we see in our and there's the condensins, help to bring those together. I think these are out of order. Those condensins should have been the other place. Cohesions and condensions. You know, the cohesions help to bring those sister chromatins together during replication, and then the condensins help those chromosomes condense, and then you get separation at ana or telophase between the 2 sister chromatins. And this is the E. coli nucleoids. And this shows you those loop domains of the E. coli chromosomes, which look very similar to the rosettes that we see. And then that's it for this chapter, I guess. Any questions?
So there was where I forgot the term. This protein that breaks a link on the DNA.
Topoisomerases, or
Relieve the twist. Does that mean that the one side of the DNA that's still linked can rotate about?
yeah, basically, so the topo type 1s do that, right? But it's not like it just rotates, because you still have this protein. So, but basically, and we're missing the first half of the figure. Right, but at this point, we have this open conformation where you have the topo type 1 that's covalently bound, and it's broke the 1 chain. The other chain can then kind of wrap around it, and then you get the link reformed, basically. But it can unwind those supercoils that develop, you know, when you have to either replicate or transcribe. Because when you separate these 2 strands of DNA and migrate up it, you develop this overwound and underwound DNA that have to be compensated for. Otherwise, this won't be able to move. You build up so much strain on one side or the other, right? Does that make sense?
Yeah, I'm trying to imagine one side of that DNA, I guess, twisting around and not breaking.
Well, they're you know, covalent bonds that aren't pi bonds. So you get rotation around those phosphates, right?
So they have free rotation?
Yeah.
Okay. When we talked about, there's another class, DNA replication in bacteria, where it's a ring, they talked about how, you know, they're just kind of, it's like pulling apart 2 rings, sort of. Now, I wasn't sure how that would work.
You mean like the clamp loader, or the clamp, the DNA, the structure that holds the DNA polymerase complex onto the chromosomes while it's replicating?
No, I'm thinking more high order than that. Like, when we're duplicating our DNA, you know, we separate and get that little bubble, and then the bubble kind of travels to the end.
And you get the two new strands that are paired with one old strand each, right?
But with DNA that's just, you know, just
Circular.
circular DNA, you know, and you get the bubble, and the bubble travels and meets on the other end, and now all of a sudden you have two rings. I was just thinking how, being helical, how that would work, how you can just pull them apart. It seems like it would get tighter and tighter on the other end.
Well, yeah, that's where the topoisomerases come in.
At some point, or maybe multiple points, there's just little breaks and they just spin around each other.
Exactly.
Like adapters for the phone cords that let it spin and that.
Yes, exactly. Which I never owned those, I always should have. So,
Well, put the phone down.
and that enzyme that typically does that is called DNA gyrase, and it's a topo type 2, where,
So if you break one of the chains, you can have free rotation of the other.
but for the DNA gyrases, typically you're breaking both chains, and it's an ATP dependent process. And it passes one chain, you know, one double-stranded chain directly through the double-stranded chain on the other. So it's how you, it's not like they have the free rotation like the topo type 1s have. For DNA replication, they're DNA gyrases. This is the type of topoisomerase that you have. They're type 2, where it breaks both chains in one strand and then allows a second strand just to unwind through that double-stranded break.